Welcome students. Today we are going to throw light on double fertilization. It was S. Nevashchin in Russia who showed that in ovules of Lilium mortagone and Fertilaria tenila, both male gametes from the pollen tube penetrated the embryo sac, whereas one of them fused with the nucleus of the egg, the other fused with the polar nuclei floating in the central cell, initiating a second fertilization event. The results of this work were presented in August 1908 to the botanical section of the meeting held in Russia, and the full paper appeared as few months after the meeting. Thus, reverent credit is due to Nevashchin for his legendary discovery of the two fusion events during fertilization in flowering plants. The phenomenon observed by Nevashchin was also independently confirmed in Lilium mortagone and Lilium pyrenacum by L. Guignard in 1899 in France. Since the discovery of double fertilization, the cellular and molecular mechanisms involved in this unique reproductive process have remained rather enigmatic. Direct cytological observations have been difficult because the dynamic interactions between the gametes takes place deep in the maternal tissues. It was only in mid-1960s the electron microscopy studies in cotton showed that gamete interaction and fusion takes place at a location where both male and female gametes are devoid of a cell wall. These cytological descriptions were repeated in various species. The design of methodologies allowing in vitro fertilization in maize led to physiological studies suggesting the conservation of calcium signaling during fertilization in plants and in animals. Species from the genus Tornea produce protruding female gametophyte, allowing direct observation of the double fertilization process. The flowering plants are the most diverse group of land plants and largely dominate all terrestrial ecosystem. Flowering plants have evolved a unique fertilization process called double fertilization. In double fertilization, two sperm cells fertilize two female gametophytic cells, the egg cell and the central cell. After fertilization, the embryo develops from the fertilized egg cell and the central cell gives rise to the endosperm, which nourishes the embryo for its development. These two fertilization events are tightly controlled and takes place in a coordinated manner to ensure successful embryogenesis. Unlike animals and lower plants such as bryophytes, lycophytes and ferns, flowering plant sperm cells are immotile and are delivered to the female gametophyte by pollen grains. The pollen grain consists of two sperm cells inside a vegetative cell. After pollen deposition on the stigma, the vegetative cell elongates and the pollen tube enters into the ovary to deliver the sperm cells. A successful double fertilization depends on: number 1, proper guidance of the pollen tube to the unfertilized embryo sac. Number 2, release of the two sperm cells towards the egg cell and the central cell. Number 3, recognition and fusion between each pair of gametes. And number 4, fusion between gamete nuclei and zygotic activation. During the past few years, identification of gamete-specific genes and promoters made it possible to mark gametes, allowing gamete transcriptome analysis and in vivo imaging of double fertilization. Now, mechanism of double fertilization. First, pollen tube guidance. Directional pollen tube growth to the female gametophyte. Preceding fertilization, the pollen tube needs to be guided from the stigma to the embryo sac embedded in the ovule. Previously, it was suggested that the pollen tube may be guided by only a single chemical cue emanating from the ovule. However, models have indicated that this distance is far too long to be governed by a single gradient and may require several consecutive cues in the transmitting tissue and along the ovarian wall. The growth of the pollen tube appears to be governed by extracellular matrix components and arabinogalactan proteins respectively. On the stigma, pollen grains hydrate and germinate to initiate pollen tube growth into the transmitting tissues of the ovary. The pollen tube then emerges on the surface of the placenta and is guided to grow onto the funiculus, which connects the embryo, sac and placenta. The pollen tube eventually reaches the micropyle of the embryo. Remarkable advances have been made in our understanding of micropylar guidance. It was first demonstrated experimentally that the synergid cells of the female gametophyte primarily govern micropylar guidance in Tornea forneri. In Arabidopsis thaliana, MYB98 is a transcription factor expressed preferentially in synergid cells. The MYB98 mutant shows defects in the organization of the filiform apparatus of synergid cells and micropylar guidance of pollen tubes, demonstrating that proper function of synergid cells is essential for micropylar guidance. Several attempts have been made to identify pollen tube attractants secreted by synergid cells. Many MYB98 dependent synergid specific transcripts were identified, including those encoding cysteine-rich polypeptides (CRPs) that are secreted to the filiform apparatus. Whether these secreted CRPs are required for pollen tube micropylar guidance in Arabidopsis thaliana remains to be determined, but pollen tube attractants were identified in Siamese and Tornea. The maize gene ZMEA1 encodes a polymorphic small protein, and the ZMEA1 GFP fusion protein is detached in the cell wall that surrounds the synergid cells. Knockdown of ZMEA1 affects the entrance of the pollen tube in the intercellular space of the micropyle. In vitro analysis showed that the predicted mature ZMEA1 protein can attract maize pollen tubes directly, further supporting the idea that the ZMEA1 protein is the attractant for micropylar guidance. In Tornea forneri, LURE1 and LURE2 encode CRPs, which belong to a subgroup of the defensin-like gene superfamily and are expressed highly in the synergid cells. LUREs are secreted from the synergid cells to the filiform apparatus and morpholino knockdown prevents proper pollen tube attraction. In vitro pollen tube attraction assay demonstrated that LURE1 and 2 indeed attract Tornia pollen tubes, confirming that LURE1 and 2 are truly the attractants for micropylar guidance. LURE homologs from Tornia concolor are expressed in synergid cells but are poor attractants for pollen tube of Tornia forneri, suggesting that LUREs might participate in the prevention of cross-fertilization between species. Arabidopsis synergid cells also express and secrete defensin-like CRPs to the filiform apparatus, and further analysis will be necessary to understand whether defensin-like CRPs secreted by Arabidopsis synergid cells are also involved in micropylar guidance. Recently, Gama-aminobutyric acid GABA was found to be one of the possible attractants that is produced by sporophytic tissues. In Arabidopsis, a sharp increase in the concentration of GABA between the septum and the inner integument cells near the micropyle of the ovule have been observed. In the gynoecium of pollen pistil incompatibility 2 POP2 mutandis of Arabidopsis, GABA levels are very high as a consequence of reduced GABA transaminase expression. However, these plants still have a steep septum to integument GABA gradient. Pollen tubes from POP2 plants grow at normal speed through the styler transmitting tract of both mutant and wild type plants, but only in the mutant ovary they show aberrant growth. There they fail to adhere to the ovule and do not enter the micropyle. Wild type pollen tubes grow normally in POP2 ovaries. POP2 pollen tubes are hypersensitive to GABA and suggest that they are unable to grow towards the high GABA sources in the POP2 ovary. However, the pollen tube attracting activity of GABA could not be reproduced in vitro, suggesting that together with GABA, additional molecules are needed. After arriving at the micropylar end of the female gametophyte, the pollen tube stops elongation. This is followed by pollen tube rupture and sperm cell release for successful double fertilization. Now, we will discuss sperm delivery, when and where. The lack of high-resolution in vivo methods to analyze sperm dynamics during fertilization prevented an understanding of how sperm cells migrate, and many hypotheses arose to explain the cellular mechanisms of double fertilization. However, rapid advances in technology and methodology, such as septotemporal high-resolution confocal microscopy coupled with cell-specific fluorescent markers, now allow the process of double fertilization to be dissected in much greater detail. Once the pollen tube reaches the micropylar end of the female gametophyte, its growth arrest and release of the two sperm cells takes place by rupture of the pollen tube tip through interaction with the synergid cells. In vivo observations suggested that degeneration of the synergid cell takes place at the time of pollen tube arrival in Arabidopsis or at pollen tube rupture in Tornia. However, the mechanisms responsible for the transport of the two sperm cells remain unclear. It was proposed that the two sperm cells are released into the degenerated synergid cell and are transported to the gamete fusion site by an active mechanism involving the cytoskeleton. An alternative hypothesis proposes that the two sperm cells are transported passively by an cytoplasmic flow of the pollen tube content discharged between the synergid and the egg cell. After sperm cell release is completed, the two sperm cells apparently become wedged between the central cell and the egg cell and do not change position until gamete fusion. If sperm cells are deposited in synergids and transferred to the female gametes by cytoskeletal elements, a pause after an initial pollen burst should be observed. Hence, live imaging observations indicate that the cytoplasmic flow by pollen tube rupture is sufficient for the sperm cells to reach the site of gamete fusions. This finding is consistent with the observations in Tornia, and this may indicate that the sperm cell delivery by cytoplasmic flow is conserved among flowering plants. The pollen tube cytoplasm could be injected between the egg cell and one synergid cell, creating a channel for the delivery of sperm cells between the plasma membranes of the synergid cell and the egg cell. The end of this channel reaches the contact between the egg cell and the central cell plasma membranes, where sperm cells remain immobile until plasmogamy. Synergid cell degeneration would be a secondary event resulting either from a specific signaling mechanism or from the mechanical stress caused by the burst of pollen tube cytoplasm. Now the question arises, do dimorphic sperm cells prevent polyspermy in double fertilization? Successful double fertilization relies on two independent gamete cell fusions. One sperm cell must fuse with the egg cell and the other with the central cell. The two sperm cells are released at the site of gamete fusion simultaneously. If one sperm cell is able to fuse with the egg cell while the other is able to fuse with the central cell, coordination of the two fertilization events is secured and polyspermy is prevented. Plumbago xylenica produces dimorphic sperm cells which express different sets of transcripts. Each sperm cell shows preferential fertilization to the egg cell or the central cell depending on its identity. Arabidopsis, like most flowering plant species, produces isomorphic sperm cells that cannot be distinguished by gene expression so far. However, only one of the two sperm cells is associated in closer vicinity with the vegetative nucleus of the pollen. This association involves the outer endocytic membrane that surrounds both sperm cells. It is thus possible that one sperm cell receives specific signals from the vegetative nucleus that would determine its capacity to fuse preferentially with one female gamete. Therefore, it has been an issue of debate whether sperm cells are functionally equivalent. The question of sperm functional differentiation was initially tested in Arabidopsis mutants that produce multiple egg cells or a single sperm cell. Mutants Eostre and Retinoblastoma related one generate two egg cells, which can be fertilized by both sperm cells delivered by a single pollen tube. Furthermore, mutants chromatin assembly factor 1 and cyclin dependent kinase A1 CDKA1 produce a single sperm cell per pollen grain, which shows the ability to fertilize either the egg cell or the central cell. Only a single sperm cell formed by translational inhibition in the male germ cell shows preferential fertilization to the central cell. Although these studies supported an equivalent functional identity for both sperm cells, they were not conclusive because they did not involve wild-type sperm cells and wild-type female gametes. To end this controversy, Hamamura and his group in 2011 used a photoconvertible fluorescent protein to mark each sperm cell differently and examined the preferentiality of fertilization of two sperm cells delivered by a wild-type pollen tube to a wild-type ovule. Both sperm cells in either position fertilize the egg cell and the central cell with a similar probability, demonstrating that Arabidopsis sperm cells are functionally equivalent. The equivalence of Arabidopsis sperm cells raises the issue of polyspermy block. Circumstantial evidence has suggested the presence of polyspermy blocks in plants. Arabidopsis mutants, tetraspore and retinoblastoma related one also indicated the presence of polyspermy block in the egg cell. Live cell imaging in wild type has provided support to these results. The two plasmogamies takes place simultaneously within less than 1 minute, and in spite of simultaneous fusions of the sperm cells, they do not observe two fusion events with the egg cell or with the central cell, because the two sperm cells are equally able to fuse with either female gamete. These observations suggest that polyspermy is prevented not only in the egg cell but also in the central cell. It remains unknown what cellular and molecular mechanisms prevent polyspermy in the egg cell and the central cell, and whether there are any differences in polyspermy block between the two female gametes. Dear students, this is all about double fertilization. Thank you very much.